Hello, I'm Shantanu Jadhav, and I'm Associate Professor of Neuroscience Psychology and also in the Wolin Center for Complex Systems at Brandeis University. And I'm a PI of a systems neuroscience lab here, and we are investigating the neural mechanisms underlying learning, memory, and cognition in the mammalian brain.
How did you first become interested in neuroscience?
And in terms of my interest in neuroscience, I would say, it was kind of a bit of a circuitous road. I have a multidisciplinary education and background. So I'm originally from India and I grew up in a very small town in India. And since when I can remember, I always wanted to be a scientist. It's something that had always been in my mind.
Really?
Yeah, I was. Yeah, funnily enough, right? But I was fascinated mostly. I think at that point with the mysteries of the universe, and I had this fascination with stars and astronomy and physics. And used to read biographies of scientists and some of them like Marie Curie, Feynman, Stephen Hawking. His book was very popular with me. But Marie Curie's biography is still my favorite book, and she's my scientific hero. So I had this really romantic view of scientists, like we are doing scientists do important work that really give us new knowledge, and we are uh, scientists are really pushing the boundaries of human knowledge and human civilization. So I always had this view.
Mhm.
uh, and I actually came from a family of doctors. And so, doctoral engineering, being a doctoral engineer, was a natural path from where I came from. But since I was interested in science, I got this opportunity to go to a fairly well-known institute in India. It's called the Indian Institute of Technology in
Mhm.
Bombay, and I chose to study engineering physics because I was very interested in physics at that point, and I thought I wanted to go on and be a physicist as I grow up. But as an undergrad, thankfully enough, we had enough of a diversity that I was able to take a couple of courses on genetics, and that got me fascinated about biology. So that led me to do actually summer in India in a neurogenetics lab. And then I got the very good opportunity to work as a undergraduate researcher in a lab here in New York in Cold Spring Harbor. They have a very nice undergraduate program. And I somehow accidentally ended up in a neuroscience lab for that summer. And once it was a systems neuroscience lab, studying audition. And once you kind of get introduced to neuroscience, it just hooks you, right? It feels to me it was the mysteries of the brain are the biggest mysteries that we have. Apart from the mysteries of the, there are mysteries within us rather than in the universe. And so I felt like, no, this is what I want to do. Neuroscience, I want to understand the brain, about how we make sense of the world around us. So then after my undergrad in engineering physics, I spent a year gaining some experimental experience back in a lab in India, in Bangalore. And then I applied to grad schools. I was again lucky enough to kind of get interviews in a few places on the West and East Coast and visit, and ended up in graduate school in neuroscience and the West Coast, at UCSD. So,
And how from
hmm.
that training during your undergrad and then grad school did you come to your current position as a professor?
Yeah, that's I must say it's a fairly maybe a standard path. So I was in UCSD as a graduate student, and as an engineering background student, engineering and physics, I was very interested in sensory systems at that point. I think a lot of engineers really like sensory systems because it's in some sense like an engineering focused problem. So I started studying the somatosensory system in a rodent whisker lab in Dan Feldman's lab. My previous experience, which was back in India, was actually the effect of stress on memory, which is very different. But that kind of convinced me that I didn't want to do purely computational work. I wanted to do experimental work because I thought it was important. And so I studied essentially how somatosensory information is coded in the barrel cortex in rodents, and in particular, I studied texture coding of texture information. And when I was in UCSD, my advisor moved to Berkeley my last year of grad school, so I moved with him. uh, and his wife who's also a neuroscientist, so they both moved. But I finished up my degree in UCSD. Then I stayed on as a postdoc there, and as I was doing a lot of behavior work and in vivo physiology, I started doing in vivo physiology in my graduate lab, which was mainly a slice physiology lab. And I thought that the cognitive questions to me started to become more interesting because I was doing a decision making task. And so I really wanted to get involved in memory and decision making and cognitive process are really these internal mental processes, right, which had been the realm of psychology for the long time, but we need to gain an understanding of how the physical brain gives us these abilities. So then I went just across the bay from Berkeley to UCSF to Lauren Frank's lab for a postdoc, where I studied hippocampal mechanisms of memory. And I started also studying the hippocampus and the prefrontal cortex, which is the executive or decision making region of the brain. And there I had we made a few discoveries about hippocampal replay and memory. And when on the faculty job market, I had a few offers, and I decided to come to Brandeis, which is a fantastic neuroscience program and environment. And so have been here for the last 8 years now.
Nice. So let's talk about your research lab. Can you tell us what your main goal or main questions are?
I think what I like to study is what I think are the big algorithmic mysteries of the brain. Like, yes, there are mechanistic mysteries also, how the brain implements computations. But there are some things I think we just don't have a good idea about is in terms of cognition. So we study the circuits that are distributed across multiple brain regions that allow animals and us to learn, to store and retrieve memories, and to plan and make decisions. And if you, I think you will agree with me, possibly the audience will, that memories are really a key to our sense of self, right? Key to our sense of self, our cognition. It drives everything that we do. And there's really something different about our brain's computation versus some other, say, artificial systems, although AI is now making strides in that direction. Right? So we study these questions in both health and disease, and I'll touch on this a bit later about the relationships between those two, because one of our long-term goals is hopefully to develop novel targets and interventions that can rescue cognitive deficits in neurological disease, which is kind of a big mystery. So since we are interested in learning, memory, and decision making, and you can often study these questions at many levels, like genetic, molecular, your immunologist, cellular, neural, and then networks of neurons, and then the whole organismal level. So I tend to take a holistic level that we eventually need to make a link across levels, but also our knowledge at the level of neural dynamics at which are seen in the level of ensembles or neural populations, we really consider a missing link that can bridge across what we know about genetic and molecular processes and also what we know about behavioral phenomena. So that's the link that we're trying to make at the synaptic and cellular level. How, when we start looking at hundreds to thousands of neurons distributed across brain regions, are there any emergent properties at this level that arise which can explain cognitive and behavioral phenomena? And can we also identify how perturbations of this phenomena, either to experiment or when they get affected in disease, right? How does that lead to cognitive deficits and not just memory disorders like, say, dementia and Alzheimer's, but also disorders like schizophrenia and autism, which are really disorders of cognition.
Yeah, very interesting.
Mhm.
What are some of the projects that are ongoing in the lab?
Yeah. So first, maybe I should describe. We use rodent models to ask these questions, and we use behavior and high density simultaneous electrophysiological recordings in many regions, as well as real time detection and manipulation of circuits and activity patterns to make causal links. And so we have had like several seminal findings that provide some fundamental insights into mechanisms underlying cognition, and we are expanding on those now. As I mentioned before, we have principally studied over the past few years the interaction between what is the brain's episodic memory hub, which is the hippocampus, and the brain's decision making executive hub, which is the prefrontal cortex. And we have asked questions about how do these two regions or circuits in these two regions work together to learn, to store memories, and then eventually to retrieve these memories to make online decisions. So a couple of our main findings I should mention first. We have mainly used spatial memory models till now, although now we are expanding and have expanded quite a lot. So using spatial memory models in which animals have to learn spatial rules and tasks in order to get rewards, this is goal directed decision making. We have found this process of awake and sleep replay, which is synchronized across the memory and the executive hubs, and which you have shown is necessary for learning, memory, and decision making in the mammalian brain. So this is really a state dependent processing in which when the brain is offline, so during online processing, we see this phenomenon called place cells in the hippocampus, which are very well known. It's like a Nobel Prize winning discovery. So this is like a map of the space in your brain, which is a cognitive map, a GPS. But what we found is during offline states, when animals pause in environment, it had been previously seen in sleep, but in the waking case also, we see an internal replay process which reactivates the same activity patterns that we saw when the animals were experiencing things or exploring things. So this replay process is thought to be one of the cornerstones of memory because it has all of the features of it. Like it's is the reactivation of a pattern of activity that was present during actual experience, right? And
Mhm.
so that's what we typically define a memory to be. But this internal replay process is an extremely fast time scale process, and this we have shown that it mediates an internal dialogue between the hippocampus and other cortical and subcortical regions, and allows animals to learn from past experience to form memories. So what actions led to reward, for example, or maybe even negative memories, and also use these memories then to retrieve these memories to make decisions about what to do next. And in this process of doing this, we have also kind of discovered that there are these multiple time scales of computation for cognition. So at the behavioral time scale, typically that's how we think when the brain processes information, right, in real time. But these replay processes and some other processes that are mediated by different rhythms that we see in large scale neural populations, we see that they have distinct computational regimes at different time scales.
Okay.
These have been our main findings. And so I think of this as extremely critical in terms of just how the brain computes.
Mhm.
They just compute in real time, but we also need to tap into this internal dialogue processes and then link this individual phenomenon to different aspects of cognition. So some of the experiments that we are following up on that now is we have more expanded behaviors where we are doing complex cognitive tasks which can be spatial or non-spatial. We also have some sensory cue guided decision making tasks, and we are looking in the hippocampus and prefrontal cortex again to see how these circuits can underlie this complex cognitive tasks using similar principles. We are also
Mhm.
looking at neuromodulatory systems like the dopaminergic VTA system, the cholinergic system, to see how these systems modulate the different states, both during waking and sleep.
Okay.
Looking at the effect of sleep on memory itself is a big target for us. And I should mention, so the other part which is looking in disease is a one of the big changes over the last couple of labs in is we have a big thrust of trying to look at disease models. uh,
Mm.
so because we look at these regions which are often implicated in several neuropsychiatric disorders, like for Alzheimer's is the
Mhm.
hippocampus, for schizophrenia the prefrontal cortex and the hippocampus both. And also have personal reason for this. I have two children, and my older son is actually on the autism spectrum, so that got me really interested in trying to look at autism spectrum disorder models. So the past 2 years, with funding from the Simons Foundation, which is one of the major funders of autism research, we have started looking at cognitive processes in autism models as well as wild types to look at both of these in parallel, right? And especially to look at things as social learning, social observational learning. So we
Mhm.
have these behavioral paradigms in rodents that can tap into these. And we are using our tools of high density physiology and closed loop optogenetic perturbation to investigate, okay, what are the neural mechanisms that allow organisms to do this complex social cognitive tasks, and also whether we can rectify these in these models.
One question that came to mind about when you were mentioning the replay systems, and
Yes.
I'm wondering if you've been able to identify any triggers for that, and if you know what the drivers of those replay systems are.
Yeah, no, that's a great question, and I should probably have Slow down a little bit so you could ask the questions as I go along. So the replay process, so far, has seemed to always been a very stochastic process. So it was first observed in sleeping animals, in sleeping brains. So from psychology literature, we always knew that sleep is important for memory, right? Like that has been well known. So when animals sleep in slow wave sleep, which is non-REM sleep or non-rapid eye movement sleep, the hippocampus just seems to be naturally in a state where there are this lot of offline state where there are a lot of sharp wave ripples, and it's
Mhm.
exquisitely coordinated with this thalamocortical spindles and slow oscillations. So when that was first observed, people thought of it as a mechanism for coordinating hippocampal and cortical circuits and subcortical circuits in order to make memories more permanent, so to consolidate memories. In the waking case, when this was observed, it's also actually controlled by a state change, because when the animal goes from an exploratory mode to a non exploratory mode, like consuming reward or grooming, essentially not paying attention to the stream of sensory information coming in, that's when we see that the brain goes in this other state, which is this offline state, and hippocampal ripples dominate. So we know that it's controlled by this neuromodulator later called acetylcholine. So when we are active and alert, this neuromodulator is high, and when we are not as alert and more internally processing, this neuromodulator train goes low. But it also kind of emphasized the importance of this downtime when we are not always looking at our phones or continuously paying attention to the external information. There is a computation going on in our brain, which is extremely important for memory.
What do you think happens when people don't have a lot of downtime? Like now we're very connected to our phones, and
Hmm.
I'm guilty of spending way too much time on it. So it's always like I'm always active in that way.
Yeah.
What do you think happen when those downtimes are affected?
Yeah, it is actually, yeah, recently somebody tried to give me a talk on this, and also there's probably important effect of stress and just our normal abilities, right? So this downtime we think is extremely important in the for the formation of long-term memories. One thing you might have noticed right away is, even I noticed with myself, is our attention spans go down when we are
Mhm.
continuously used to streams of information, right? It's hard to attend internally to a task for a long time. You're MD PhD students, as a medical student, you probably read a lot of books, right, which requires you to sit for like 1 or 2 hours.
Too many.
Too many, yes, yeah, for a long time. And I used to do that also, but I noticed that the more I'm engaged, the more my attention span is impaired. Also, our the effect on our short-term memory as well as working memory is, it's kind of documented. We have a lower working memory if that happens, so more harder time to keep our information in memory. As far as the longer term effects on memory, like maybe a decade later or so on and so forth, that we will soon find out, I think, in the longer run, because there's not enough research. It's actually well known for children, for example, children who are exposed to screen time for since really early in development, it really affects their development. They might start speaking later, affects their attention processing. So those things are actually well documented.
So downtime is important.
So take some time off and smell the flowers. Yeah.
Anything else that you want to add regarding your ongoing work?
We of course have a very wide definition of what we do in terms of, when you say neural mechanisms of cognition and behavior, that's very broad, right? And I'm very really welcome people to the lab who bring different perspectives as well as a diversity of thoughts, and who want to take the lab in a different direction. We have some boundaries around stuff that we can do, but the good thing about Brandeis also is we are a very collaborative environment here, and also we are in the Boston area, so I have collaborations with many people both within the institute and across. I'm in general interested in questions related to behavior and cognition, and would welcome diversity of perspectives. So uh, do not hesitate, I would say, people to contact me if you're interested in a particular question, right, related to information processing in the brain or memory or any kind of cognitive processes. That's something that you would like to pursue with the tools that we have in our disposal.
Yeah, it seems like there is a little bit of everything for anyone that is interested.
Yeah.
type of work that you do.
Yeah, and maybe a couple of things I should also mention, which I didn't before, is we are also doing some computational work, modeling work, because if our goal is to eventually understand how everything works together and integrative understanding, you have to be able to reproduce it, right? If you understand the principles of something, and that is something that has been typically difficult to do, and one of the way to get is to eventually build complex computational models of things. And one of our findings also has been on related to chemosensory information in our memory system, which is fairly new. We are actually looking at gustatory processing, taste processing within the lab, olfactory processing. So basically, everything that is relevant to the brain is something of interest to me. Although we have a focus, but we are open to expanding that focus also.
What is one thing that people can expect to get from or learn from their time in your research group?
Yeah. So that's a great question. And there are a couple of different things, and this goes to a little bit to training and mentoring style also, like what kind of training. One is of course the technical aspect of it, but the other is, I would say, conceptual aspect, or being a scientist or a scientific thinker or even a leader, right? In that sense. So I really feel, having been in my lab for 8 years now, so it's not that long, but I've had grad students who have come and gone through the lab. I have postdocs who have come and gone, and we recently went through a lot of graduate students leaving and new ones coming in. It was very bittersweet for me because the students I really identify with, right? Because the first PhD students were the ones whom I started the lab with. So I feel like people really define you, like me as a PI, and my success depends on people's success in the longer run. And my impact on the field would be through also training and mentoring people. So I try to have a very kind of open mentoring style, in the sense what my goal usually is to try to spur people's interest and act them to do their best work and eventually be leaders in what they are doing, both in the thought process and the techniques. And my goal as a PI is just to remove their barriers, right? So they can do their best work. So if they have any hurdles, I'm there. I have an open door policy. Of course, I have regular meetings as well, but I keep an open door policy. And I would like them to think of a particular question that they want to answer. I have an adaptive style. Some people want to bring their own questions or are more open to exploring. Other people need more guidance. So I would sometimes give them a few options of things to look at. They can go look at the literature, but it's really the drive to answer an unknown is something that I want to spur in people in my lab, get them excited about it, because that's when they're going to be motivated to answer it. So hopefully that is the main thing that they will take away from the lab, to always have that interest, that burning desire or motivation. And in terms of techniques, we do rodent behavior, we do in vivo electrophysiology, we do optogenetics. We also do a lot of data analysis and computational work. I actually teach a class in data analysis also, which I think is a very important thing, regardless of any branch of science now, like even life
Mhm.
sciences, right? You have to learn how to deal with big data. So those are also tools that you will acquire.
What drives your work? Or in other words, what is something that you're excited about your work or the field in general?
So as I said, neuroscience is just something that hooks you, because I feel, we often call it biology's last frontier, or this is the century of the brain, right? There are so many fundamental mysteries about how our brain works, and how it essentially supports our sense of self, that are key to answer. And once you kind of accept the possibility that is the physical circuits in the brain that are underlying all of the complex things that we can achieve, that we do, those to me are just exciting questions in their own right. And of course, as one person working on a project, you're often targeting one small question in relation to the bigger question, like the bigger picture. But that's what all PhDs are about eventually, or all
Yes.
projects. But the fact that you can make a dent in the unknown, right, and push our knowledge forward is what excites me, and in particular in an area like neuroscience, where there's a lot to understand about the brain. I'd mentioned before that it's the algorithmic mysteries. What I mean by that is we do not really know some fundamental principles of, say, how memory is incorporated in the computations that the brain does. Can we understand something fundamental apart from the details of the different circuits and how something particular happened? Can we learn something broader? Um, and this is just an example, like, could we design an AI which can reproduce the brain's capabilities, right? Not that that is what we want to do, but I think if you understand a system well enough, then you're able to reproduce it artificially, right? And I think we lack that level of understanding of the brain yet. And I also feel that it's important to get at this understanding in order to target disease, like, can we actually help people with regards to this? So, memory is, as I said, is often impaired in several cognitive disorders. That is disorders of cognition. For example, in schizophrenia, you'll have hallucinations, right? So,
Mhm.
what does that mean really? That you means you're imagining something. So there's something in your circuit. We have circuits of imagination also, right, which are linked to our circuits of memory. To have an hallucination, which means that process is impaired in our brain. So we need to understand the natural the natural process, the native processing in the brain, to understand how these perturbations in disease occur in order to target it. So, pharmacology is, of course, one way to go to try to target disorders, but we are hoping that we can also add tools at the systems level, which can be targeting neural dynamics using, say, closed loop techniques, which can be used in concert with pharmacological treatments to treat many disorders. So, the possibility that we can make advances in this is what excites me most, both from, like, I would say, a pure science perspective, and also from an applied or a clinical perspective.